Hello, and welcome to a Taconic T-Onco webinar entitled Molecular Imaging, given by Dr. Xavier Tizon from our French oncology partner company, Oncodesign. First, I want to introduce myself as the host of today's webinar. My name is Megan McBride, and I work in Taconic's Global Business Development Team, where I am responsible for genetically modified models in the areas of oncology and immunology. I will be your moderator for today's webinar. Before we get started, I'd like to address a few commonly asked questions for this type of presentation format. First, today's presentation will be available at the end of today's session. The link to the presentation will be emailed to all attendees at the end of the week. Also, we will be answering questions at the end of the presentation. If you have a question, type your question in the control panel, which should be at the bottom right of your screen. Other attendees will not be able to see your question as they are sent directly to the presenter and will be answered at the end of the session. If you cannot see the control panel, just mouse over the top of your screen and a drop-down menu will appear. If your question is not answered at the end of today's session, one of our scientists will respond to your question within the next 24 hours. We will then email you with our response. If you are experiencing technical difficulties, raise your hand in the control panel and our staff will assist you. Let me introduce today's speaker. A trained biomedical engineer, Xavier Tizon completed his PhD in medical image processing at Uppsala University, Sweden in 2004. His research focused on semi-automatic segmentation methods for the analysis of whole-body magnetic resonance angiography images. He then returned to France, where he spent a year working as a postdoctoral researcher and teaching assistant in the medical imaging lab at the University of Burgundy in Dijon. Since June 2006, he has been the head of the imaging lab at Oncodesign, where he routinely uses MRI, SPECT, and PET to assess the efficacy of anti-cancer therapies in rodent studies using rats and mice. His team works in close collaboration with several research institutions to develop and validate imaging biomarkers of pharmacological activity dedicated to preclinical and clinical studies. We hope you enjoyed today's webinar entitled Molecular Imaging.
Thank you, Megan. So I'll be talking about molecular imaging or pharmaco-imaging as a tool to enhance drug development in oncology. So first, an outline of this presentation, I'll talk a little bit about how difficult this is to develop an oncology drug and how imaging biomarkers can help with that. I'll go through a series of examples, first talking about antibodies and labeling molecules to help in drug development. Then I'll talk a little bit about how imaging can help in quantifying tumor proliferation. And then last example series will be about imaging tumor vascularization using MRI. And I'll draw some conclusions at the end. So first, some words about how difficult it is to develop a novel therapy in oncology. First, you have to deal with how different the drug can be efficacious on different subsets of patients. So you have to think about selecting your patients during your drug development. You also have to think about early evaluate the efficacy of your drug because most of targeted therapy bring biological activity through cytostatic activity and not tumor shrinkage. Also, lots of drugs fail in late clinical trials because of toxicity and safety problems. You have to think of it very early in the development, and there is a real need for earlier indicators of this toxic activity. And there is more problems even. Translational results from small animals and from in vitro experiments are very difficult to translate from small animals to humans. You also have to think about managing variability and heterogeneity between and within patients. And you also have to think very early of how your drug can be combined or the therapeutic strategy changed because it's quite obvious with what we know now that monotherapy won't be very successful. So one possible solution with that is the use of imaging biomarkers. During the development of your drug, you ask a series of questions Is the target present in my system? Is the drug in contact with this target? Is this target engaged? Does it produce inhibition or activation of this target? Is there a biochemical consequence of this engagement? Is there any downstream biochemical change that is a consequence of that? Is there any biological change happening a little bit later and at a larger scale? And at the end, maybe the most important question, are these changes related to a clinical endpoint? So this is a schematic summary of the drug development process, and you go through a series of steps from in vivo target identification to clinical evaluation, from the molecule to the patients. And the benefits that those imaging biomarkers can bring you is that they can allow you to non-invasively and serially monitor your tumor. They give you localized information about the drug effect. They also give you a possibility to early evaluate tumor response. And I'll show you also that those biomarkers are translational in nature and can be included in early clinical trials. So for each step in this drug development, early drug development process, there are imaging modalities that are available to answer these questions. So why imaging? Basically, the main advantages of imaging is that it's non-invasive. It allows for repetitive measurements. It's an in vivo snapshot of a biological or biochemical process. One also of the very important advantages of imaging is that it's possible to assess tumor heterogeneity and different localizations simultaneously. It's translational. I'll talk a little bit more in detail about that. The drawbacks are that it costs quite a lot of money to perform imaging studies, but there are proof now that it can help you in building more efficient strategy for later clinical development. Of course, expertise is required, but it's the same with other kind of biomarkers. And at this stage, at this point in time, there is still insufficient validation of those biomarkers and still too much site-specific protocols. The first series of examples will be basically about labeling your molecules to help your drug development, with mainly examples drawn from antibody research and using a technology called single photon emission computed tomography, or SPECT. And I'll show you examples of target expression and biodistribution and efficacy monitoring. First, just one word about the technology itself. It's quite simple. You have a radiopharmaceutical or radio tracer that you inject to your patient or your animal system, and the radiopharmaceutical binds to its target inside the body, and then you use the machine to build the 3D biodistribution of the radio tracer in the body. So the way this kind of study is built is that first you have to think about your synthesis methodology. This is an example based on the research that's going on in Dijon through the pharmimage project that I will talk about a little bit later. We have created a new labeled trastuzumab, a new labeled antibody with indium. And first you have to devise a strategy to do that. Second, you have to be sure that the properties of your antibodies are preserved from this labeling. So you have to test in vitro that the affinity and other properties of your antibodies stay in an acceptable range. And then you're ready to inject to animals. And this is an example of images on mice. And you see that we've imaged 1, 2, and 3 days after label antibody administration in a tumor model that is overexpressing the target of trastuzumab, HER2, and we see a nice accumulation in the tumor through the 3 imaging time points. We most of the time confirm this with biodistribution studies after dissection of the animal. So 3 steps: first radiochemistry, then radiopharmacy, and then injecting to animals, which we can call radiopharmacology. The first research example I will show is on using this kind of technology. It's indium trastuzumab, again, that is used to study the the effect of an anti-VEGF antibody on HER2 expression using an HER2 overexpressing cancer model again. So in this study performed by very recently, we can see a decrease in trastuzumab uptake in the tumor that is induced by treatment. And with this study, they have shown that this is mainly due to changes in vascular permeability and blood perfusion using what can be called support experiments. In that case, and using these technologies, support experiment can be autoradiography, so it's the equivalent of nuclear imaging, but ex vivo, and immunohistochemistry using the targets or the mechanistically involved targets of the compound. So just a small clinical example related to this problematic, using again a labeled trastuzumab and patients with HER2 overexpressing tumors. And the goal of this study was mainly to diagnose tumors, and they have shown, you see clearly on these images that there's a large liver metastasis that is on top. This is a CT image of a patient, and on the bottom line you see the superimposed SPECT image showing a large lesion that has been pointed out by this tracer. So the advantage of using this tracer in that case is that you can identify not only the primary tumor, but also the secondary tumors throughout the body. Unfortunately, in this study, they have shown that there was a low detection rate, and this is caused by technical limitations of SPECT in humans. That's why I'm going to switch to another nuclear imaging technology called PET, for positron emission tomography, and show very briefly these results on labeled antibody again. It's a different antibody. It's bevacizumab targeting VEGF. In this study, they have measured the difference in uptake for this label antibody. That was the consequence of treatment, and treatment with a drug called everolimus, which is an inhibitor of mTOR. And they have shown that after treatment, the uptake of labeled bevacizumab went down as a consequence of the treatment. So the use of PET to study antibodies has been more and more in the recent years, because the technology has allowed to use, for example, zirconium-89, which has a half-life of almost 80 hours, which is well suited to study antibodies. In that case, again, they have used support experiments, namely immunohistochemistry, to confirm in vivo imaging results. I'm going to close this part on labeling molecules with just a few words about our imatinib research project. We started this research project 3 years ago, and the goal of this project is to use a technology that we have acquired that is called nanocyclics, and that is a technology based on cyclase kinase inhibitors that we are building into PET tracers. So the idea is to use those very specific kinase inhibitors to build a uniformed labeling strategy and use those using the different kind of targets that we have available to build new PET tracers. So the second series of example will be on the positron emission tomography technology using fluorotimidine, which is another tracers. I'll show applications of efficacy and the biological process that will be studied is tumor proliferation. So very briefly again, I'm sorry for the technical problem here. The guy seems to be upside down, but it's basically the same principle as with SPECT imaging, except that instead of having one sensor, you get two sensors. The consequence of which is that the technique is about 100 times more sensitive than SPECT, and you get a better spatial resolution. So a few words about FDG first, which is fluorodeoxyglucose, which is the most used radio tracer in the field of PET. It's a very good tracer. It's highly sensitive, but the problem is that it targets glucose metabolism, and it's not very specific of tumor-related processes. Fluorotimidine, on the other side, is a structural analog of timidine, which is almost exclusively found in DNA. It's not incorporated in DNA, but it's trapped in the cell due to phosphorylation, which makes it a good tracer because there is accumulation of the tracer in the cell. And it's currently under a clinical trial for quantitative and non-invasive imaging of tumor proliferation. So it's thought to be useful for diagnosis, for treatment monitoring, and also for monitoring of cytostatic therapies inducing cell cycle arrest. So in this example of using FLT, FLT is used to try and understand the efficacy of everolimus, which is the inhibitor of the mTOR, in a resistant ovarian tumor model. So in this study, they have shown that the PET quantities, the things that you measure using PET, mainly the SUV for standard uptake value, goes down as early as 2 days after treatment start, which is actually showing changes before changes in tumor volume. Changes by 1/3 2 days after treatment start and 2/3 after 1 week. And in this case, they showed good correlation with immunohistochemistry as well. So this was a positive example of the use of fluorotimidine to measure early efficacy in small animals. I would like to say a few words about the development of biomarkers of efficacy for the mTOR inhibitor everolimus. In 2 articles published in 2010, Novartis described how FLT-PET may not be the right tool to measure efficacy, and that a biomarker with a high selectivity for a given pathway may be difficult to use because of the complexity of this pathway. Mainly, they conclude that one of the main reasons why this compound shows efficacy is its anti-angiogenic and antivascular properties. In this case, it may be useful to combine molecular imaging techniques with functional imaging. We think that this is exactly what pharmaco-imaging is about, choosing the right tool given what we know about a compound. So the last series of examples is about, can be applied to research on small molecules, multi-targeted tyrosine kinase inhibitors, and it's using a technology called dynamic contrast-enhanced magnetic resonance imaging, and it can be and has been used in a multiple application from prediction of response to resistance and optimization of combination. What we are seeing is things related to how well the tumor is vascularized. So very briefly, what the technology, how the technology is working. We perform dynamic imaging, meaning we take an image every few seconds, and at some point in time, we inject a contrast agent and measure the concentration of this contrast agent over time. At the end, we can pinpoint the tumor or a specific region of interest inside the tumor, and then do pharmacokinetic modeling and getting a few parameters. One of the most known parameter is called K-trans. So I'm going to present one example of some work we have done a few years back. This is data of tumor volume over time at several time points in 3 groups using a multi-kinase inhibitor on nude rats and on a subcutaneous lung cancer model. So we can see here that there is a dose-dependent effect on tumor volume, and at the first imaging time point, namely 2 days after treatment start, we can see that there is this dose-dependent effect on the K-trans parameter, which is not yet visible on tumor volume. So it gives us early indications of the efficacy of the compound. This has been confirmed by immunohistochemistry, the support experiments I was talking about. So basically what we have concluded is that the effect that we have seen may be due to vascular normalization inside the tumor. The compound has followed its development, and it's now in phase 2 with this imaging technique used as a PD biomarker of efficacy. So this is a paper published in 2011 with the clinical data on this compound, now called tivozanib. The last example I will show is from a clinical study using bevacizumab and the same technology, dynamic contrast enhanced MRI. What we are seeing here is the different parametric maps that are given by the technique, and these are morphological imaging through time. So it's from 4 hours after treatment to 12 days after treatment. So what's happening through time is that there is a gradual reduction of the tumor enhancing fraction, meaning less and less of the tumor volume is vascularized, and we also can see that the tumor is shrinking quite rapidly. So what this study brings is that we get multiple parameters in this study, at least 4 in that case. And what O'Connor and et al. Have seen is that some of these parameters gave information on tumor function. Those changes are seen early and are rather transient. And some other parameters give information on on vascular structure, and those happen a little bit later and are more permanent. So in this case, it's really interesting to see that not only one measure has given information, but several measures at the same time has given information on what's happening in the tumor. So we have seen what's available with imaging tool through the oncology drug development. There are applications that are available right now as target validation, biodistribution, optimization of treatment schedule, and finding of the biological active dose. You can also, of course, evaluate the tumor response to treatment, but more applications are being seen and are very interesting for the future, related to on and off target toxicities, studying combination of compounds and cell trafficking. And using multimodality, you can do one or two or more of these in one experiment. The challenges are also numerous. First, there are technological barriers, tracers that have to be developed, imaging devices that have to be better than what they are now, and you also have to work on data analysis methods. The second set of challenges is about integrating these imaging data together and with other biomarker data like genomic data. The third is about validation. I talked a little bit about this at the beginning of my presentation. Most of these imaging techniques have to be standardized, both on the data acquisition phase and the image analysis protocol. They also have to be qualified. That is, it has to be shown that they are useful for a given application in the clinic. In all those cases, one of the biggest difficulties in using imaging methods is that you need a multidisciplinary team. And the only interesting question at the end is, of course, will imaging biomarkers be useful in the clinic? We'd like to think of pharmaco-imaging as a model, and in that model you have to think of several things. First, a tentative drug mechanism of action, which represents your hypothesis. Then you have to choose a relevant animal tumor model. You have to choose a given modality and an acquisition protocol and an analysis strategy to try and interpret the data that you're seeing. Most of the time, it's very useful to have in parallel a validation experiment with gold standard methods. And at each step of this process, you have to have translation in mind. It's very important to try and build your experiment so that it can translate to humans. To achieve this goal of our pharmaco-imaging model, the second research program that we've initiated a few years ago is called pharm-image. And this is actually a map of the area we are in Dijon, and this is us. We basically represent in this consortium the experts in pharmacology and in animal models, but as I said, you need multidisciplinary team, so we need more experts, we need chemistry, we need image processing, and that we have built from the research team around us. We also need radiochemistry, we need expertise in biomolecules, and finally, we need teams that have knowledge of what's happening in the clinic. And this is all pharm-image. This is a group of research labs and small companies building around the concept of pharmaco-imaging. So I like to conclude by saying that in pharmaco-imaging too, the best is yet to come. We are starting to understand why cancer is such a complex disease, and that oncogenesis and the effect of treatments are the result of a balance between very complex and very robust biological phenomena, like cell proliferation and angiogenesis. We think imaging has a role to play by combining information from multiple sources to build an imaging phenotype of the tumor. And that concludes my presentation. I'll be happy to answer any question you may have. Thank you.
Thank you, Dr. Tizon. That was great. We will take questions at this time. Please go ahead and type your questions into the chat box. Only the presenters will see them. If you cannot see the chat box, just mouse over the top of your screen and a drop-down menu will appear. I have one question that just came in. How much time does it take to develop a new imaging tracer?
Actually, it takes quite a lot of time. We have examples, for example, the development of FDG and FLT has been going on for decades now. But it's mainly because the process was not as standardized as it was for new medical entities, for drug candidates. So nowadays, it should take a little bit less time, mainly because tracers are developed like new medical entities now, and there are ways to perform clinical trials using tracers.
Okay. We have another question. How many animals are typically included in an imaging study?
Well, that depends on the modality and the aim of the study. Basically, there is one limitation related to that, is that because of the fast-growing nature of tumors on animals, we need to image all animals the same day. So depending on how many animals you can image in one given day, this gives you the constraint on how you build your groups. For example, in the technologies, using the technologies that I've shown examples from, for example, PET with FLT or DC MRI, you can have from 4 to 6 animals and 3 to 4 groups in a given experiment. If you perform other kind of experiments, for example, one technology that I haven't talked about is bioluminescence imaging. Then it takes only a few seconds to a few minutes to image one given animal. So you can have 15 animals per group and 5 to 6 group, and you can perform all your imaging experiments within one given day.
Okay. Do you also use statistics to determine the number of animals you need in each group?
Actually, it's not a requirement for preclinical studies, but we're beginning to see some of our clients requiring objective sample size in these kind of preclinical experiments, sample size measurements.
Okay.
So we are mostly limited by what I talked about, how many animals can be imaged within one day, but the objective way of doing that and the methodology of preclinical trials is rising, yes.
Okay. We have a question. Do you have clinical trials in the pipeline with any of the molecules you've developed?
Not yet. No, we are not at this stage yet.
Okay. Can you talk a little bit about the use of bioluminescence imaging and how that fits in with the other modalities you talked about?
Yeah. I didn't talk about it mainly because there was not enough time. This is a very useful technique. It's very broadly used in small animal experiments. There are tens and maybe hundreds of devices throughout the world that are used to perform pharmacological experiments with. It's mainly used in the field of oncology to study tumor growth through transfected tumor models that you inject either subcutaneously or orthotopically, for example, in the abdomen cavity to see your tumor growing. It's very sensitive. You can image a few thousands of cells and you will see them. It's high throughput. It's lower cost than other technologies, but using conventional techniques, it's at best semi-quantitative, and this is quite a big drawback. The latest machines are beginning to use tomographic techniques and are more quantitative, but the basic, let's say 2D bioluminescence image is only semi-quantitative.
Okay. Can you talk a little bit more about the cost and time savings that can be expected when you use the same animals in a longitudinal study? And also, what is the impact on the precision of the data when you use the same animals versus when you have different groups of animals for each time point?
It's a little bit difficult to give documented answers to those questions because those pertain more to statistics than imaging. But in the case of, let's say, a follow-up of a tumor growth with PET, for example, you can think of an imaging experiment with 6 animals per group, and you can image your animals during the whole growth of the tumor, meaning maybe over a month or so. So you have 4 or 5 imaging time points in this month. And, well, if you compare this kind of design with the design in which you would have to euthanize each group of 6 animals at every imaging time point, then the multiplication is not very difficult to make, and you would use as many more imaging animals that there are imaging time points.
Okay. Have you considered incorporating contrast ultrasound?
Yes, of course. Actually, there will be a webinar at the beginning of 2013 dealing exclusively with angiogenesis imaging. And in this webinar, I will, of course, talk about all the available technologies to image angiogenesis. And in that case, ultrasound, contrast ultrasound is a modality of choice, yes.
Okay. Thank you. We're running out of time, so I'm going to wrap up now. We will be sending out a link to the slides so that everyone will have an opportunity to download them or view the webinar later. We do hope you have enjoyed today's webinar. Thank you to Dr. Tizon. This concludes our presentation. Thank you.